Thank you very much to Africali, the African Alliance for Allergy and Immunology, for giving me this platform and inviting me to speak about the first 1,000 days, the early life interventions for allergy prevention. I'd like to thank our sponsors as well for providing this platform. My name is Professor Kirtika Patel, and I have no conflict of interest presenting this work. I come from the east of Africa. In a country called Kenya, which is renowned for its wildlife, safaris, scenic landscapes, long-distance runners. It has a rich culture and vibrant coastal cities. So in Kenya, I come from the western part of Kenya, called Eldoret, and it is in the Rift Valley, almost near the equator, and it is very well known for the city of champions. All the marathon runners that you see from Kenya come from this place. It is also the city of champion researchers, it has been cited. So I come from Moore University, and Moore University is in the, it houses 15 schools, and one of the schools is the School of Medicine. Moore University is situated in a rural part of Eldoret, while the hospital in which the School of Medicine is situated is in the semi-urban, semi-rural settings. So this is the hospital, and this is our School of Medicine. Moore University is built on 3,000 acres of land, and you can see it's in the beautiful Rift Valley. This is an aerial shot of the university, and this is the hospital, and this is the School of Medicine. So welcome everybody to this webinar, and today we'll be discussing a critical window of human development, the first 1,000 days. It's not just a phrase, it's a biological reality where the immune system is highly malleable, and mistakes or disruptions during this short window can set a child on a lifelong trajectory towards allergic disease, while the right exposure can protect them for decades. So the first 1,000 days is also referred to as DOHAD. It's an acronym for the Development of Origins of Health and Disease, and it spans from the period from conception to the second birthday of the child. And you can see there is exposure from intra to extra-uterine environment, and it's a very time-sensitive window to rewire the infant's immune system from allergy-prone to tolerant. The seeds of most of the diseases sown, most of the diseases included allergy, are sown during the period of rapid growth and development. And the first 1,000 days are critical period of a child, and it's also called the golden interval. So how is the 1,000 days calculated? So the period consists of 270 days of pregnancy plus 365 days till the infant is 1 year old, and then another 365 days from 1 to 2, and that makes a total of 1,000 days of life. Our philosopher earlier said that, yes, the history of man for the 9 months preceding his birth would probably be far more interesting and contains events of greater moment than all the 3 scores of the years that follow it. And even in UNICEF's homepage, you'll find that the first 1,000 days of life is the time spanning roughly between conception and one's second birthday. And this is a unique period where the foundation of optimal health, growth, and neurodevelopment across the lifespans are established. And the period consists of 270 days of pregnancy, and it changes at birth to 1 year, where the child is fed with breast milk or formula milk, and then he's weaned off, and solid foods are introduced later on. And from 1 to 2 years, the child grows through an intense growth process. So in the first 1,000 days, the body undergoes growth and development at the highest rate during a human lifetime. And during that first critical 1,000 days, the right nutrients are very important for the development of the digestive system, the brain, the gut microbiota, the metabolic system, and of course the immune system, which we will be focusing on. Nutrition is very important because it's an integral part, and it could have a short-term health effect and a long-term health as well, and it can span into adulthood, and it can lead to obesity, diabetes, stunting, allergy, and heart disease, and we'll see how it's done. Let's unpack the first 1,000 days, which is a window of opportunity. And today we will, through a strict mechanistic lens, look at it. This period represents a critical therapeutic window. During this phase, the neonatal immune system exhibits high development plasticity, and it is influenced by the environment, nutrition, and microbial exposure. And it interacts with the child's epigenetics to determine lifelong allergy susceptibility. The biological window of vulnerability and plasticity is a critical period for epigenetic marking as well, where the DNA is methylated and histone acylation. It interacts with the child's epigenetics to determine lifelong allergy susceptibility, and it is a transition from a sterile environment in the maternal fetal symbiosis to dense microbial colonization. Allergy March is not a new thing to this group, but it is a progression of allergic diseases that start in the infancy and develop sequentially through childhood, so from eczema to food allergy, and then the other airway disorders. So the atopic march it leads from food allergy all the way right through asthma. And the mechanism is the skin exposure, which causes the allergy, and the gut exposure creates tolerance. So there are 3 phases in a neonate's life. So the phase 1 is prenatal, where the maternal diet is very important. Phase 2 is when from birth to 6 months, where the gut microbiome health and skin barrier protection is important. And phase 3, which is from 6 to 24 months, is early allergen introduction protocols. This atopic march is a current crisis, and the childhood food allergies are increasing in up to 10% of infants now globally. And it's a chain reaction, and there is a progression we've seen from eczema in infancy, predictably leads to food allergies, asthma, and then rhinitis later. So our clinical goal should be to stop eczema early to break this chain. And there is a dual allergy theory the gut versus the skin, where the skin exposure is sensitization, which is the beginning of the chain reaction. So the disrupted skin barriers will let the allergens in and triggering early allergies, while gut exposure will create tolerance. So eating the same allergen that would have caused the sensitization when taken orally, the gut immune system accepts and it is safe and creates tolerance. So there is an immune shift. Early oral exposure switches the body from an allergic response to a protective immune response. So here we have the evolutionary hypothesis based on firstly, and it is showing the epigenetic programming from preconception to pregnancy. Preconception to pregnancy, there is an influence of the maternal diet and external environment. The maternal diet could be the nutrition the mother is taking, the calorie intakes, the vitamins, the environmental would be stress level, pollutants, physical activities, the geographical location, and these have inputs into the epigenetics of the baby, and it can trigger and turn off various genes on or off in gene transcription, and it shapes the tissue growth and fundamentally metabolic settings and permanently alters the programming of the fetal immune, metabolic, and endocrine functions. So essentially, the fetus takes a biological snapshot of the mother's current world to prepare itself for life after birth. So then, after that, there is programming of the, if there is a match between the maternal and infant diet and environment, to the appropriate immune, metabolic, and endocrine response. However, if there is a mismatch between maternal and infant diet and environment, there is increased risk of allergy, autoimmunity, obesity, metabolic syndrome, and cardiovascular disease. For example, if the mother's body signaled a resource scarce environment, the child is born into a resource scarce environment, and this child develops appropriate immune, metabolic, and endocrine. However, if there is a mismatch, it can result. So, if the mother is aggressively to store fat and energy, and being born in a modern Western environment where there's a high calorie, nutrient dense diet, this will result in a mismatch and will lead to obesity. Lead to allergy and obesity. Okay, the evolutionary hypothesis also states that the womb preparation, a mother's gene and environment interactions during pregnancy prepares the body for the outside world, and the risk is lifelong. So a little bit of immunology, I'll try to simplify as much as possible. But just please have your attention for just this slide. That the naive T cell or T cell has, in the presence of various cytokines, can mature into a Th2 cell or a Th1 cell, a helper Th1 or a helper Th2, or a Th17 cell. And all these cells are regulated by the T regulatory cells. Now, what is important for us in pregnancy is that we need the suppression of the inflammatory cells, which are the Th1 and Th17, to avoid pregnancy loss. and proliferation of the Th2 and the T regulatory cells, which will help the pregnancy to succeed and tolerate. So the fetal-maternal immune paradox. In utero, the fetus requires an obligated Th2 skewed environment, and the neonate is born with default, which is hyperactive Th2 bias. And this Th2 bias is important for fighting parasites, which means nothing when the child is in utero, but it is a driving force behind allergic reaction, asthma, and eczema. So an unprogrammed pool of naive T cells are also present along with the Th2. So at birth, so during pregnancy, there is Th2 cells dominant, but there is now, after birth, a sudden shift in the immune balance. So now we need the Th1 cells to proliferate, because you will now have, you've come in an environment which has a lot of microorganisms, and you need your Th1 cells to protect you. And they produce also the inflammatory cytokines that endanger the fetus. And the Th1 response triggers a maternal cell-mediated rejection. So this is the components of during pregnancy, the maternal immune system faces a unique challenge. It must tolerate the fetus that contains 50% of foreign genetic material from the father without leaving the mother entirely vulnerable to infection. So there has to be a balance between the regulatory cells and the inflammatory cells. So, so in order to do that, there is the Th2 response and increase of regulatory cytokines, and a decrease of Th1. And the fetus will be protected, and there will not be any rejection because of the regulatory T cells. So let us look at this chart, which a flow diagram, which Professor Warner has given us in the fetal origins of allergy and potential nutrient intervention to disease prevention. And it shows how maternal factors and early environmental exposure interacts over time to shape a child's lifelong development. So here, this diagram shows from conception, birth to 2 years, and how the maternal allergy, diet, microbiome, composition, preconception health, and pregnancy conditions, a fetal immune system that is naturally biased towards Th2 and Tregs pathways. Following birth, the maternal allergy, diet, microbiome, and preconception, along with the maternal environment during pregnancy, has an effect on the maternal and fetal genes and gene expression. And the fetal immune ontogeny will be Th2 Treg based. And at birth, the maternal diet, microbiome, and environment during lactation has an effect on the postnatal environment, depending on whether the baby is breast or bottle fed. The infant's microbiome, route and time of allergy exposure, along with the postnatal immune ontogeny at 2 years. And if there is a balance between Th1 and Th2 regulatory cells, then the baby will be non-allergic. However, if there is an imbalance between Th1 and Th2 regs, there will be allergy sensitization. This is another algorithm of gene environment asthma and COPD, and it shows how specific genetic traits interact with early environmental exposure to drive different forces of chronic lung diseases. So there are two pathways. So allergy genes, you could have allergy genes, and the allergen, and with the exposure of the allergen, you could have sensitization. Or the second pathway is you could have the ADAM33, which is a structural gene which affects fetal lung morphogenesis, and in the presence of various pollutants like smoke, paracetamol exposure, tobacco, you could have an airway dysmorphism. And now, if you have sensitization, you could have an allergy but not asthma. You could have Th2 airway inflammation and asthma. And both of these, allergy sensitization and allergy airway dysmorphism, could converge and give you the Th2 airway inflammation causing asthma. And the airway dysmorphism, we know, without allergy could give the infant wheeze, and the infant could have non-allergic asthma or COPD. So there is a gene environment interaction. Early gene environment interaction, which drives the two completely separate respiratory pathologies. So there is a structural and alteration or immune changes. The first 1,000 days represents the early life origins of allergy sensitization. The antenatal factors could be maternal, genomic, epigenomic, metagenomic, diet, and environment. And we'll look at the maternal factors in a minute, maternal environmental factors in a minute, and in the postnatal period, which is infant, microbiome, diet, route, and timing of allergen exposure. And they are critical because you have to balance the Th2. And failure to downregulate the neonatal Th2 response is associated with a higher risk of allergic sensitization. Studies have shown that the prenatal maternal environment is very important, and it can be modulated. So the exposure here, there is a farmer exposed to the stable dust, diverse and bacteria, and animal environments, and this helps the fetal and neonatal immune response, lowering the chance of atopy later in life. So this study was done in the US, and a lot more studies have been done after that. Exposure to live farms, early live farm, helps maternal activities while pregnant, were strongly associated with reduced risk of atopy in adults. And then we are aware of the South African study, Professor Levin and his team, and that also beautifully show how the early childhood and maternal exposure to farm animals in rural settings protected against allergic condition, and they also gave us the risk factors that were in both the farm and the rural and the urban areas. And it showed how distinct environmental and life factor directly influences the child immune development. Latest study from Munich by Pagani's unit showed that the farm cow shelters specific gut bacteria, and these gram-positive bacteria, they generate various metabolites, and these metabolites, when inhaled by humans, they bind to receptors on the airways that suppresses the inflammatory processes. So here now we have the proof that this biological mechanism accounts for 2/3 of the farm-associated protection against childhood asthma, hay fever, and atopic eczema. This farm effect is now seen where it's now visible from the cow to the barn air, the bacteria, the metabolic products that the bacteria produces, the receptors in the human, and it protects against asthma. And this new finding can be used even by lab researchers to decipher the molecular and cellular mechanism of farm effects. And it now opens up a prospect of developing probably a drug that mimics the farm effects without the need for children to spend time in barns. The postnatal pivot, educating the naive T cell, now the infant. How can we help the infant? So the birth triggers an immediate need to activate the Th1 arm now. And the microbial exposure is very important to stimulate the Th1 arm. The Th1 will drive the production of various cytokines, and the goal is to reach homeostasis between the Th1 and Th2, the inflammatory Th17 and the T regulatory cells. So the birth of a child is an immunological boot camp. So at that time, the baby's naive immune system must be trained quickly, and the shift from Th2 to Th1 is very important. And if this shift does not occur, then it could lead to the atopic march, eczema, progresses to food allergy, asthma, and allergic rhinitis. The postnatal lymphocyte ontogeny and the atopic march, the key mechanisms and the key players are the bacteria, or the pathogens, or the microbes, and they have on their surface molecular patterns which are recognized by the infant's T cells, and this drives the differentiation of the naive T cells into Th1 clones, and this balances the system. It also preempts the dendritic cell to produce IL-12, so that dendritic cells is the antigen-presenting cell, and it leads to Th1 differentiation. And failure to this transition from Th2 to Th1 will result in B cells that will switch to IgE, which we know is very important in allergy, and this will lead to clinical atopy. There are various external factors, and microbes definitely shape it. So it depends on the birth mode, whether it was a C-section or a natural delivery, whether the infant was breastfed or formula milk, whether there was antibiotic use in the mother and the infant, which shapes the gut microbiota composition, and also the induction of T regulatory cells. They give education of mucosal immunity and the maturation of adaptive immunity. So the microbial colonization, if it was a vaginal delivery, will expose the infant to various bacteria like lactobacillus and bifidobacterium, which are very important for populating the gut. However, with the C-section, they will have only the environmental bacteria. And this disturbed dysbiosis, a disturbed microbial community delays the signaling through pattern recognition receptors on the intestinal epithelium, altering the early gut maturation. And this stalls the microbial succession, the dysbiosis, and the microbial diversity peak rapidly by the age of 2, resembles an adult gut. And this looks complicated, but it's a very nice diagram to show the various milestones. So pregnancy, the neonate is protected, and it depends on the maternal diet and the maternal microbiota as well. And we do not see a lot of neonatal microbiota at that stage. However, the immune system is developing, and you will see the innate cells coming up at birth, depending on whether it's a C-section or a natural delivery. If it's a natural delivery due to the fecal and vaginal microbiota, there's transplantation of these microbes into the baby, while the C-section will not be able to colonize the gut with these natural microbiota. And then lactation has an influence. If the baby is breastfed, the microbes from the mother will be passed to the baby, while formula milk will not have that. And the immune system is now maturing from innate, and it will now have the immature cells proliferating, including the NK cells. And then at the age of, and then at infancy, when the child is now weaned off, or it is now introduced to solid foods, the microbiota develops. And it's amazing how a 2-year-old and an adult have a similar microbiota. In terms of immune development, from innate, it moves on to adaptive, and the child has a very solid immune system by the age of 2. In the first 1,000 days, the child's immune system progresses, and because of the presence of microorganisms. How do these bacteria stop allergies? So actually, these bacteria talk to our immune cells. These bacteria, they utilize the human milk oligosaccharides, which is present in the breast milk. And these HMOs do nothing for the baby, but they are the food for these bacteria, and they colonize the mucus layer and protect the gut epithelium. They also stimulate the production of T regulatory cells. We know that the Th1 and Th2 are our foot soldiers, but the T regulatory cells are very important, and they are generals calling for peace. They are the ones who tell the immune system that this peanut protein or pollen is not an enemy, so stand down. So they regulate the immune system, and therefore it is very important to have these T regulatory cells. And the primary goal of earlier microcolonization is very important. And the induction of T regulatory cells are the principal immune pacifiers. So these bacteria, they modulate the dendritic cells, which are the antigen-presenting cells, and these cells are important to generate the T regulatory cells. And T regulatory cells produces anti-inflammatory cytokines, which downregulate the Th2 and the inflammatory pathways, and therefore establishing systemic oral tolerance to begin dietary antigens. These bacteria eat up the dietary fiber and break them down into short-chain fatty acids. And these acids, short-chain fatty acids, are important in turning off the on and off the pro-allergic genes. These fatty acids feed the cells lining the gut and welding them together tightly, and strengthens and prevents the leaky gut. Along with the bacteria, there is also the immune, the breast milk also is seen not just as a nutrition, but also as an immunological toolkit, and it's a powerful immunomodulator because it has secretory in IgA, which coats the gut, and it neutralizes pathogens. The HMOs consist of sugar that is feeding the bacteria. The milk also has maternal IgE allergic complex, and it carries tiny foods from bound to antibodies from the mother, giving infant a safe preview of the dietary world. And this teaches the infant immune system tolerance. So the burden of food allergy is increasing, and it is approaching 10% in infants. And we have seen it is a sequential progression driven by epithelial barrier dysfunction. And our clinical goal should be to interrupt early onset of dermatitis to halt respiratory and systemic sequelae. So there are 3 phases. Phase 1 is the prenatal intervention, that is conception to birth. And the recommendation is no elimination diet. Restriction of major allergens during pregnancy does not prevent childhood allergies. Diet diversity. The pregnant mother should eat a highly varied diet containing natural allergens. And they should also have a diet with omega-3 and vitamin D. Phase 2 now is postnatal, and it is from birth to 6 months. And here the gut-skin axis is very important, and the benefit of breast milk, and also dysbiosis risks, allergies, and the skin protection is very important. So aggressive moisturizing of infants' eczema prevents the allergen to enter the skin. Therefore, and this has been seen, backed up by lots of research. And one of a big study was the LEAP study, learning about peanut allergy, where the infants with severe eczema or egg allergies were given peanuts starting from 4 to 11 months. And there was a reduction, 81% reduction in allergies. So the early introduction trains the immune system with highly adaptable 1000-day view. And this is a window of tolerance. You have to introduce the allergen around 6 months, but not strictly, not before 4 months. And these are the 2026 guidelines. And one-time taste does not work. The allergen must be eaten weekly to maintain protection. And it is also backed up by another study, the EAT study, the inquiring about tolerance. And here they evaluated and said that the introduction of 6 sequential food shows the success with adherence was high. The food allergens plummeted by 67% and 75% reduction in egg and peanut allergy. So there is a window of tolerance. The optimal physiological window opens at the development readiness around 6 months, but not before 4 months. And the dosing and frequency are very important. Tolerance is an active, dose-dependent process requiring consistent chronic exposure. The environmental exposure and the hygiene hypothesis plays a big role in developing our immune system. And the old friends hypothesis, where we had said the human beings evolved along with the soil, the animals, and the gut microbes, is now coming out, and it would urbanization and sensitization has reduced our exposure to environmental exposure. So the immune system is like an advanced AI. It needs massive data sets of harmless environmental microbes to train its algorithms. And without this data, it will begin misidentifying harmless proteins as deadly threats. Also, there is collateral damage if the infant is exposed to antibiotic and antacids. So early life microbiome is very important, which could be destroyed by the use of antibiotics, and even antacids would harm and the gastric acid, and it will prevent the breakdown of the proteins which are necessary for oral tolerance. The evidence-based clinical practice guidelines are 4-pointed protocol where you preserve the microbiome, barrier management, active oral tolerogenesis, and environmental diversity is important. The guidelines also says that no routine pre-screening, skin prick testing before food introduction is unnecessary. There's a high-risk triage. Reserve allergy screening only for infants with severe uncontrolled eczema. And the 3-day rule is very important. So introducing one single allergen at a time, waiting for 3 days, and before trying a new one. And warning for formula feeds, you should avoid giving casual brief cow's milk formula in the first week of life to breastfed babies, as it will cause allergy sensitization. The core areas for early intervention is from fetal. This chart has been taken by a study by the Warner study. Prenatal to birth, the primary target is fetal immune programming, and the intervention is maternal diet, prebiotic intake, and optimal vitamin D. From infancy, 0 to 6, we need to target the gut microbiome and skin barrier. So the intervention is exclusively breastfeeding and treatment of eczema. From 6 to 24 months, there is oral immune tolerance, and the intervention would be early and consistent introduction of diverse food, solid foods, and major allergens. During preconception period, maternal obesity and maternal nutrition is important. Weight loss intervention, smoking has an effect. Healthy, balanced diet with less meat and more fruits and vegetables, and optimal vitamin intake, and no allergy avoidance during pregnancy as well. No allergy avoidance. Medications to be avoided if possible to maintain the maternal microbiome. And during delivery, C-section to be avoided, bottle feeding to be avoided, breastfeeding to be encouraged in the neonatal period. It is important for the infant microbiome to proliferate. And the key clinical takeaways is a shift in mindset, transition immediately from defensive avoidance to active oral exposure. And it is a combined strategy, the next maternal nutrition, aggressive allergy care, and early feeding for the best results. And there are strict timelines. This biological window closes around age of 2 as the immune system is now mature. So the first 1,000 days is highly sensitive metabolic and immunological crossroads. You need to optimize maternal nutrition, modify early life exposure, management of the epithelial barrier, preserving the microbiome, early oral tolerance, and embrace nature. We can actively prevent atopic diseases rather than merely treating symptoms. I looked at the Kenyan guidelines, and it was on the first 1,000 days, and it's a pretty good guideline encouraging foods during pregnancy, and then maintaining those healthy habits and breastfeeding, and also a diet rich in vitamins with fruits and vegetables was encouraged. The future of allergy research, we need to focus now on shifting from single treatment tests to looking at the whole picture, the real world picture. We should look at the diet, the pollution, the medication, and the germs. We need to map the biology as well. Genomics and epigenomics are very important because the genes can be turned off and on due to the environmental factors. Metagenomics, which is the gut bacteria ecosystem, and the metabolites that they produce, that is the metabolics, are very important. And our ultimate goal is to predict exact infant health and allergy outcomes before the symptoms ever appear. So this is a graph from the Academy of Medical Sciences UK report, and it showed that from preconception to elderly, the risk of chronic diseases increases and the resilience decreases if there is no intervention. However, if there is early intervention, you can stop and increase, you can stop the rise of the chronic health disease. Late intervention does have a role to play as well. And the public health and global initiatives. Strategies are disease surveillance and early detection, herd immunity and community engagement. Public health initiatives are vital in disease prevention. Awareness campaigns improve vaccination rates. And now there are emerging technologies, and we should use them for research to make these treatments available. However, there are challenges and limitations. Access to healthcare information, vaccine hesitation due to misinformation, and addressing these challenges require policy interventions and public education. So prevention strategies, public awareness, and early interventions are our new way of dealing with allergies. Thank you very much for your attention. I'm open to any questions or comments. Thank you very much.